Hello, everybody. Good morning and good afternoon. Welcome to today's webinar titled Fingerstick-Based DNA Collection Using truCOLLECT Solution for Whole Genome Sequencing in Pediatric and Neurogenetic Research. My name is Sydney Panilla. I am with LabRoots, and I will be your moderator for today's webinar. Today's educational web seminar is presented by LabRoots and brought to you by Covaris. To learn more, visit covaris.com. We encourage our attendees to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will go ahead and answer as many questions as we have time for at the end of the presentation. You can also use the Ask a Question box to let us know if you happen to have any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speakers for today's webinar. Paola Giusti Rodriguez, PhD, Assistant Professor, Department of Psychiatry, University of Florida College of Medicine. Elinette M. Albino, MT, PhD, MSc, Associate Professor, School of Health Professions, University of Puerto Rico Medical Sciences Campus. And Uli Toman, PhD, Vice President, Research and Development, Covaris. Okay, Uli, I will go ahead and turn it over to you first. You may now begin your presentation.
So thank you for the kind introduction and for inviting us and having us today for this LabRoots seminar about truCOLLECT, a whole blood collection kit. And I will give you an introduction before we're going to move into the real world, how to use this device and how it can be used for population genetic research. truCOLLECT is a solution, a full solution. It's not just a blood collection kit. It is designed for ease of use. That means you can collect the blood, you can stabilize the blood, you can ship it, you can store it, and you can extract the DNA in a very nice workflow. And I will talk to you. It's a versatile, decentralized, small volume blood collection device, and it adds benefits to support routine genetic testing, including oncology, clinical trial recruitment, long-term monitoring of clinical trials, and treatment, including biomarker testing. I will specifically talk today about DNA, but other biomarkers are possible as well, and we can talk about that if you are interested after the presentation. A comprehensive workflow, as I mentioned, for blood collection biomarker extraction, and it has benefits for both the user, the end user, so the person that you want the blood to be collected from, and the laboratory. It is a 50 microliter collection volume, so it's small, but you will be surprised how much DNA you get out of that. It's a simple finger stick approach, so it's avoiding phlebotomy, and there's no complication, no cold chain shipping and required because it desiccates the blood and immobilizes it in the tube, and then we can rehydrate the blood using our AFA technology, the adaptive focused acoustics, and I will show that. That's a very important technology for that, and we can extract biomarkers, even long fragment DNA. I will show you this. The alternatives is bright blood spots. They are established, but they yield low DNA quantities and quality. And of course, saliva is also a possibility, and swabs, but they all have their logistic issues and stability issues. A saliva, especially because it has a very high count of bacterial DNA, and you never know what kind of coverage you get when you do NGS sequencing. So it is designed for self-collection, ease of use, and lab efficiency. And when we did the specifications, we said we want to have it shipped after collection. For that, we had to immobilize the blood, but we immobilize it without any solid matrix. So that means we don't use paper and swab, and it's a very important thing to get good quality and high quantity DNA out of that. We stabilize it at room temperature. That's very important. So we can actually stabilize it. When I say stabilize it, is stable from -50, that's what we tested, to +60 degrees Celsius. And you can ship it at room temperature. There's no cold chain logistics required, which reduces the cost by 50 to 75%, depending on what you use. It has full traceability, very important. The sample gets collected in a vessel that is also used to do the extraction. So you will not have any cross-contamination or switching, like dried blood spots need punching, and you can mix and match during that punching and collection process. It also allows you quantitative analysis if you need it for DNA, less important. But for example, if you want to do RNA and proteins, serum proteins, plasma proteins, you will need to have a metered blood volume. So it is a solution, as I mentioned. It has a collection device and a extraction kit together with our R30 focus ultrasonicator, and that's the full solution. So you collect, you can ship, and you can extract and get the DNA out of that. It is a AFA-based rehydration, as I said, and cell lysis, and this is the most important step. If you use AFA for that, you gently rehydrate the blood, which is fully desiccated, is flaky. You lyse the cells, and you extract high quantity and high fragment molecular weight DNA, up to 96 samples per batch, and is a magnetic bead-based purification, which means this is compatible with almost every automation out there. So the solution is, number one, the collection kit, as I mentioned, and we see the device here in the upper line. You basically have the collection kit. You collect the blood. You see the blood is collected in a capillary that meters 50 microliters. Then when you pump this little blood that is collected into the device, it gets immobilized at the bottom of the tube in a proprietary matrix that is not solid. It's inert, but it keeps the blood on the bottom of the tube. After that, you cap it with a desiccant cap, which you see on the right-hand side, which dehydrates the blood, and you can ship it at room temperature. Once in the lab, very simple accessioning. You scan the barcode, rack the tube, and either store it at room temperature until you want to extract it, or you extract it the same day by rehydrating the blood and extracting it in the Covaris R230 sonicator. And then you do an automated purification or semi-automated purification depending on your batch size. So the convenience kit, the collection device, basically has a collection device pouch. So you see this in the red box. By the way, the red box is not necessarily something that you can have your own box. You can also have your own convenience kit as long as it is Lancet-based. But we basically supply it right now. You can have that as a whole collection kit, as we did in this study that you will hear about. Collection device pouch is there, the desiccant cap pouch, the IFU, of course, and then a convenience kit. And convenience kit, we mean basically the lancets, the alcohol swab, etc., what you need for Fingerstick. And the other part of it is the IFU that is in there. I show it just as a screenshot. It looks like a placemat, and it's a very simple instruction that you can use. And also the way of placing all the items there so that you have a very nice workflow in front of you. That's the device blown up in a high resolution collection device on the left. It has the blue cap and the tube, the blood collection tubes, which is metered, the 50 microliters blood into the capillaries. Then you put that thing upright, and you use the blood expeller cap, the blue cap, in order to pump the blood in there that is fully expelled from the capillary. Then you turn it, you remove that capillary holder with the capillary and toss it away, and just put the desiccant cap onto the tube that is inside the holder, and you can ship it because it is getting dehydrated at the same time. The laboratory workflow, once you shipped it by room temperature or with normal mail to the laboratory, arrival and a two-step accessioning step, you read the label, and you rack it. And the rack is provided in the kit. It's a truCOLLECT 96 home rack, and it looks like a matrix tube. When you look at that, that is correct. It is a kind of a matrix tube with a special plastic. And we have this arrayed as a 96. You can do 8 to 96 samples in an extraction that uses this shuttle in order to take all of the tubes out of the rack and into the R230 and back into the rack for capping, decapping, etc., etc. So this is all automatable, fully automatable if you want. You can also move it by hand back and forth. And this is the extraction kit, laboratory workflow. It has all of the reagents, the consumables, accessories, and again, an IFU with this. This is all done in a lab. I'll show you in the next slide how the workflow is done. It's a very simple workflow. Once the truCOLLECT tube arrives at the laboratory, you scan the barcode, populate the limbs, assemble blood tube into the 96 shuttle rack. Then you decap, either by an automatic decapper or a handheld decapper. You discard the desiccant cap, you add the DNA extraction buffer that's provided in the kit, recap it, move it to the R230, do a 60-minute AFA treatment. Very important, that gently rehydrates the blood, lyses the cells, releases the DNA. Then you decap and move the lysate to a purification plate, and then of your choice, you can either do it in the isopure, with it in an isopure, or in a Kingfisher, or you use it on a liquid handler platform. The whole thing, up to 96 samples, is done in 2 hours and 30 minutes. That means you get 96 DNA extracted in 2 hours and 30 minutes with this device. The quality and quantity is excellent. We can stabilize these the sample, the blood, and get the DNA out of this up to 6 months. We guarantee that. The yield average 1.5 micrograms per sample, which is way enough than what you need for a normal NGS analysis. It is also compatible with mechanical and enzymatic fragmentation for all of the NGS library preps that you have out there. And the DNA, because of the high fragment size, which you see in the middle, is a fragment analyzer trace. It's about 45 kb, greater than 40 kb, and up to 100 kb there. The DNA is compatible with Oxford Nanopore workflow. We have done that. I'm not going to show any data for that. But the quality, the quality and the purity, as you see on the right-hand side, is excellent. And then I'm going to close with 2 slides, because in this study that you're going to now hear from our collaborators, from Paula and Elinette, we did a full solution from collection to NGS. Basically, we collected the blood in the truCOLLECT device. We sent it via Florida, from Puerto Rico via Florida to Covaris, and then we extracted it in a process that I just showed you. And then after that, the DNA was directly used for shearing in the R230, which now you see you can use this for the whole workflow. You shear it. Mechanical shearing has many advantages, and used our library prep, which we call truCOVER library prep, which is an integrated DNA shearing, mechanical shearing, and library prep for Illumina sequencing. The hands-on time is less than 25 minutes. And the turnaround time is pretty well established and beats a lot of other kits out there, 2 hours and 50 minutes, fully automatable if necessary. In this case, we did not amplify, we didn't need to. And I show you one last slide about data, because we did all studies where we did blood and saliva samples from the same donor, converted them to libraries using the truCOVER library and also competitor kits. And then we did a comparison between the two methods, between the 2 sequencing data, and we got greater 90% concordance between those with our kit, and way less with enzymatic kits, which you see on the other one, 86, 83, and 80%. And it's not unheard of, because we know that the shearing, the mechanical shearing that we're using, is superior to enzymatic shearing, because it is very reproducible. It's not dependent on the sample. It's not dependent on the input concentration, and it's not dependent on any GC content. That's very important. I want to finish with just my part, thanking Heidi Giese, who did the project lead for this project, as well also was instrumental for truCOLLECT development. Vanessa Process, who did a lot of NGS data analysis, and then Kelly, Elena, Luca for experimental work, and also Martina Werner and Eugenio Daviso. Martina is the director and in the R&D, and Eugenio is the VP for the solutions team for general support. And I'm going to hand it over now to the way more exciting part of Elinette and Paola, and I'm looking forward to hear that. Thank you.
Hi, everyone. Great to join you today to talk about our experience collaborating with Covaris using the truCOLLECT device. So, I work in the field of psychiatric genomics, which, like many other fields in genetics, still suffers from a lack of diversity in the ancestral populations that are studied. As shown on the left panels, individuals of European ancestry are overrepresented in psychiatric genome-wide association studies compared to other global populations. This is particularly apparent for Latin American populations. And this has real clinical implications because the data that comes out of these, the studies that have different, that don't have quite the representation that they should have, ends impacting how we use the data for polygenic risk score prediction, as shown on the right. And so it has a real consequence, this lack of diversity of genetic studies. And so this is an area of focus for my group in terms of collaborating to try to correct some of these disparities in genetic studies, especially focused in Latin America. And this has real impacts across every stage of the research enterprise when we're talking about human disorders and human diseases. So, there's from how you recruit in terms of the target population that you study, assessments that are used to study them, the type of SNP array that's used to genotype samples, the reference panel that we use to look at that data, as well as the methods that are used for analyzing the data, and the functional annotation down the line. And so there's a real urgent need to address this lack of Latin American representation in psychiatric genomic studies. The lack of Latin American representation in psychiatric genomic studies has several causes. So some of the research barriers that researchers in Latin America face have to do with research capacity, a lack of collaborative opportunities, the genomics and computing infrastructure, and a lot of adequate methods to account for the complex admixture that is found across the region. And I'm showing you a graph on the right where you can see for North America, Central America, South America, and the Antilles that there's this great diversity in terms of the ancestral representation across the region, which is not uniform in the continent, and certainly not even uniform within each country. And so this means that we require a lot more samples when we're studying complex genetic diseases like psychiatric disorders across the region. And so to try to address this, I started the Latin American Genomics Consortium. I co-founded it in 2019 with some other colleagues in psychiatric genomics to accelerate psychiatric genetics research in Latin American populations and to facilitate collaboration globally, particularly focused in Latin American countries and the US. This initiative aims to not only address the underrepresentation of Latin American individuals in genomic studies, but to facilitate access to training and resources and engage in collaborations across the region. And from 2019 to now 2026, we've gone from 25 to over 200 members, representing 10 Latin American countries, the US and other global members as well. And our leadership includes 3 co-chairs as well as representatives to the larger Psychiatric Genomics Consortium. In the 6 years that we founded, we've been able to amass cohorts across Latin America and of Latin Americans in the US. We've created analytical pipelines to focus on highly admixed samples. And we have both some completed studies that are in the process of being published, as well as several other genetic studies underway to try to look at the specific genetics of Latin Americans across many psychiatric disorders. And so, for example, in 2025, we published this really wide-ranging review in Nature Genetics about focus on the psychiatric genetics in Latin American populations. And so both focus on the how common these disorders were compared to world populations, what type of genetic studies have been done, what we've uncovered, and as well as the gaps and challenges that remain doing this type of research across the region. My lab and my lab members also did a literature review focused on trying to identify what is the landscape of functional genomic studies focused on psychiatric disorders that have used samples from Latin American populations. And again, much like we see with the genetic studies, there's this big gap in terms of participation and inclusion of samples from Latin America in functional genomic studies of this type. Before I hand it over to Elinette, I think I wanted to talk about this other initiative that I'm part of called Latino, which is a network of investigators across Latin America, the US and Canada, that has been focused on collecting samples for obsessive compulsive disorder over the past 5 years. This is an image from this study where we aim to collect deep phenotype data as well as DNA from over 5,000 OCD cases, and this required this wide range collaboration with investigators, therapists, other people with lived experiences across Latin America to try to advance our understanding of the genetics of OCD. Latino has been very successful. It covered 45 sites across 13 countries. And given the number of sites and countries involved, it was really challenging to develop standard operating procedures for the shipping of samples across borders. And while we were able to stick with our timeline and collect over 5,100 cases, OCD cases across Latin America with all this phenotypic data, and saliva samples that are in the process of, DNA is getting extracted, and we're going to be genotyping it and sequencing it in the immediate future. It was very challenging getting to this point. And so this required much more complicated coordination within each of the 13 countries that participated. Many times we had to rely on local couriers or had to contact local governments and customs because saliva samples were considered a biohazard, and it required complicated shipping from the countries to the US to then do the DNA extractions and sequencing. And so, there were a lot of learned lessons in that space that were definitely a motivator for this collaboration. And also Elinette and I are both from Puerto Rico. And so while this was unfolding, we were always looking for ways to work together and advance psychiatric genomics in Puerto Rico. And so she's going to now tell you about her experience for a population in Puerto Rico using the truCOLLECT device and the work that we want to do in the future together.
Thank you, Paola. Now I'd like to briefly introduce my role and the personal mission that drives my work. I specialize in clinical laboratory and advanced diagnostics with a focus on genetic testing stewardship, and this involves developing and implementing testing algorithms, ensuring accurate interpretation, and addressing the ethical, legal, and social implications of our research. My research is centered on understanding the genetic underpinnings of neurogenetic and rare pediatric disorders in Puerto Rico, particularly autism and other related multisystem conditions. I focus on uncovering genotype-phenotype correlations and improving population-specific genomic interpretation. Ultimately, my mission is fourfold, is to shorten the often painful diagnostic odyssey for families and help implement comprehensive genomic testing as an early first-tier standard to ensure that our research is conducted with trauma-informed and ethical care for all participants. And finally, to build genomic capacity and expertise here in Puerto Rico and across Latin America. A significant part of this mission involves tackling the long and arduous journey many families face in simply getting a diagnosis. So the diagnostic odyssey is a term we use for the prolonged and often frustrating journey families endure to find a molecular diagnosis for their child. Unpublished data from my lab showed a period of uncertainty that can last anywhere from 3 to 14 years, marked by multiple inconclusive tests and fragmented evaluations across various specialties. Families are left to navigate this complex medical system without a genetic clarity. They're desperate. So our strategy is to combat this and to implement first-tier genomic sequencing, integrating both copy number variant and mitochondrial DNA analysis from the start by using a trio-based approach, sequencing the child and both parents, and adhering to ACMG guidelines for clinical reporting. We can provide comprehensive genomic clarity much earlier. So, however, as we implemented this strategy, one of our major bottlenecks remained is sample acquisition. So this bottleneck forced us to reevaluate our methods and prioritize the well-being of our participants above all else. This brings me to the core principle of our research. High-quality genomic data should never come at the expense of the patient dignity. So for our pediatric participants, especially those with sensory sensitivities, the traditional blood draw process can be traumatic. So we recognize that alternative trauma-informed collection strategies were not just beneficial, but absolutely essential to conduct our research ethically and effectively. To understand why this is so crucial, let's look at the specific barriers created by the traditional method. Traditional venipuncture or drawing blood from vein presents multiple obstacles that limit research participation and create health disparities, particularly for children with autism. The process can trigger significant psychological distress and sensory overload. It introduces logistical complexity requiring trained phlebotomists and clinical settings, which creates scheduling barriers for busy families. And this, in turn, leads to geographic disparities, and those in rural or remote settings are often excluded, mainly in our island. And finally, the sample handling itself is demanding, requiring immediate processing, cold chain maintenance, and hazardous shipping protocols. So to address these challenges head on, we initiated a pilot study with a very specific community in mind. So our pilot study focused on Puerto Rican children and adolescents with autistic phenotypes, a population that has been significantly underserved in genomic research. So our goal was to enroll children and their parents, establishing clear inclusion criteria to ensure relevance of our findings. And we established a decentralized collection infrastructure with 2 trained nurses operating across 3 community clinics in San Juan, Ponce, and Mayagüez, allowing us to reach families across the island. So you may be asking why Puerto Rico was the ideal setting for this crucial work. So Puerto Rico, for us, represents a critical gap in autism research. The population has been largely excluded from major genomic studies, and most existing autism databases are composed of non-Hispanic white population. So this has resulted in a limited understanding of genetic ancestries within the Latino community, as Paola explained. Furthermore, historical barriers, including a complex history of medical research mistrust and socioeconomic challenges, have made traditional clinic-based research difficult for us. So our goal was to address this by identifying genetic variants in this population, developing inclusive reference databases, and ultimately informing precision medicine for Latino families. We want to test a new collection method. We designed a direct head-to-head comparison. So our pilot study, we conducted a pair comparison. Each participant, including pediatric participants and their family members, provided both venous blood sample and the fingerstick sample using truCOLLECT device. To ensure consistency, both samples were collected by the trained phlebotomist. The critical difference lay in the logistics. So the venous samples require freezing and shipment on dry ice, while the truCOLLECT samples were stored and shipped in room temperature. So we then evaluate the samples based on the set of rigorous performance metrics. We assessed the entire process of 4 key metrics: the success rate of collection itself, the sample stability over time, and to ensure it was sufficient for next generation sequencing analysis, and the DNA quantity and quality compared to gold standard of venous blood, and finally the downstream NGS analysis performance, including library preparation success and the concordance of genomic data between the two methods. So a cornerstone of this study was to decentralize approach to reach communities across the island. So our collection sites, as I mentioned earlier, in San Juan, Ponce, and Mayagüez, so from northern areas, southern and western areas, provided strategic coverage from urban to rural areas, including remote islands communities often left out for research. So the truCOLLECT system was the key enabler here. Its ability to be stored at room temperature eliminated the need for freezers at each site, and the simplicity of standard mail shipping made participation feasible for geographically isolated families, significantly reducing their travel to burden. So this approach directly addresses the long-standing logistical barriers that have hindered enrollment in past studies. Families face numerous logistical challenges, including lack of transportation, time conflicts with work and school, and the difficulty of coordinating schedules for multiple family members. So these often lead to a high no-show rates for appointments. truCOLLECT here helps overcome these barriers by opening the door for future home-based collection, reducing appointment times, and creating less stressful experience that increases willingness to participate. The room temperature stability provides a level of flexibility that is, for me at least, a game changer for both researchers and families. So let's revisit the core problem we are solving, and it truly underscores the importance of my work. So this illustration powerfully depicts the reality of venipuncture for many children. The process can be fraught and full of behavioral and sensory challenges, leading to anxiety, fear, and meltdowns. So this has a profound impact on families, causing parental distress and caregiver anxiety too. So there are also technical and safety risks, such as sample loss or injury from movement. So these factors become major barriers to research participation in our study, as family may decline to enroll or drop out after an initial negative experience, reinforcing medical trauma and mistrust. So now let's walk through the entire workflow to see how truCOLLECT simplifies the process. So this diagram illustrates the parallel workflows. The top path shows the truCOLLECT process: collection in Puerto Rico, storage and shipping at room temperature, and extraction at Covaris. The bottom part shows the venipuncture process, which requires freezing and costly dry ice chipping at every step. The logistical simplicity and efficiency of truCOLLECT workflow are immediately apparent here. So let's look at the breakdown of families who participated in this groundbreaking study. So we successfully enrolled 97 participants across various family structures, including duplets, triplets, quads, and quints. From these, we successfully converted 47 truCOLLECT samples into libraries for sequencing. For our direct comparison, DNA from 15 matched venipuncture samples from affected children was also sequenced in parallel. Now, for the results, first let's examine the DNA extraction yield and the quality. The results were outstanding. We achieved 97.9% extraction success rate from 97 truCOLLECT samples. The average DNA yield was nearly 1,600 nanograms, far exceeding our target, with the highest yield of 5,600 nanograms. Only 2 samples failed. The analysis of the DNA fragments showed that the mean fragment sizes were generally greater than 35 kilobases, demonstrating excellent quality suitable for whole genome sequencing. So this high quality DNA translates directly into a successful library preparation. When we compared the DNA for sequencing using truCOVER library prep kit, we found that libraries from truCOLLECT samples had a higher average yield. You can see 94.3 nanograms versus 72.1 nanograms, and a larger average fragment size, 751 base pairs versus 681 base pairs, compared to those from venipuncture. So this indicates that DNA is robust and performs exceptionally well in this critical step. So next, we move to the sequencing data itself. A key metric for sequencing accuracy is the percentage of reads with a quality score of Q30 or higher. So at this graph clearly shows that the data points for truCOLLECT and the venipuncture are tightly clustered together, indicating no significant difference in the percentage of high quality reads. This confirms the exceptional quality of the data generated from truCOLLECT samples. So this consistency continues across other critical sequencing metrics. So while mean genome coverage was slightly lower for truCOLLECT, it was still well above the 25x threshold, considered excellent for research. More importantly, when we look at coverage across 145 known autism candidate genes, we saw excellent and uniform coverage, even across regions with difficult GC content. This ensures we are capturing a complete and accurate picture of these critical genomic areas. So, but ultimate test is concordance. So, do the two methods produce the same genetic results? Next. This is the most critical for validation. So using industry standard tools, we compared the genetic variance score from 15 matched samples. The results were definitive. We found here that concordance for SNPs was greater than 98.5%, and for indels was greater than 97%. And this is well within the expected range and confirms that truCOLLECT delivers data that is statistically indistinguishable from the traditional gold standard. So using PharmaCat tool to analyze genes that are important for drug metabolism, we found 100% concordance in the clinical reports generated from truCOLLECT and venipuncture samples. This is a profound finding for us. It means that for clinical applications like predicting a patient's response to medication, the results are identical regardless of the collection method. So this is a powerful implication for integrating this method into routine patient care. So beyond the data, the most powerful validation comes from families themselves. The data is compelling, but the human impact is what truly drives us. We heard from Maria, the mother of a 7-year-old with autism, who said, "For the first time, I didn't have to choose between helping research and protecting my child from distress." And for example, Carlos, the father of a 12-year-old, told us, "This method doesn't just advance research, it respects our children." So these testimonials powerfully highlight that this technology is not only just about better science, it's about more compassionate science. So this work will not be possible without the support of many. We would like to extend our sincere thanks to all families whose participation made this study possible, to our many collaborators and to our funding sources, including NIH. Thank you for your attention, and we are happy now to take your questions.
All right. Perfect. We have all of our speakers joining us on camera now. Thank you all for the very informative presentation. Now, before we move into the live Q&A, we do have one acknowledgement. So, Uli, I will go ahead and turn it over to you.
Yes, I didn't put it on the list because I think I have to acknowledge one person that was instrumental to getting this study done, to make the connections, and also to be the champion of the truCOLLECT. And I would like to acknowledge our exec VP, who is now moved on to more busy work, Rich Doverspike, very much. I remember when we first had the first contact with Paola at, I think it was ASHG in Boston, and we were excited about this because it was the right study at the right time. And I'm very excited about the data. I never had any doubt. Rich never had any doubt. So thank you very much. And be busy at your new assignment.
All right, fantastic. Okay, we will now go ahead and move into the live Q&A portion of the webinar. So as a reminder to our attendees, if you happen to have a question that you would like to ask, please go ahead and do so now if you have not already. You can type your question into the Ask a Question box and just click Submit. We will go ahead and answer as many of your questions as we have time for. All right, let's go ahead and dive right in here. So we'll go ahead and start with this one. It says, for studies conducted in geographically dispersed or resource limited settings, what are the best practices for ensuring chain of custody, sample stability, duration, and regulatory compliance, CLIA/CAP, or research use settings when using truCOLLECT for decentralized collection?
Who wants to take it? I can start, but I refer to to Elinette and Paola to chime in. I don't think that there is any barriers of using truCOLLECT because we are basically pursuing to have it as a Class 1 registered device, and also IVDR approval. So I don't think that there's any issues, and it is actually made for those geographically challenged sampling. There may be some barriers which I'm not aware of in countries all over the world, but that is all up to discussion. I know that at least in Europe and in many parts of the Americas, there is no barriers because it is not a biohazard. That's the good thing when you ship it. Unlike saliva, and I know that saliva, people think that it is an easy thing, but no, it has really big problems in terms of shipping from country to country. I know it just from Canada to the US, and it's a big issue, and it always hangs up at the border.
Yeah, and what I can say is that we've had experience both where we've shipped kits, like, with other studies, like we've shipped kits to people, to participants, and then they can ship them back, or the collection's done at a clinic, right, or by one of the recruiters. And in either of those will be very compatible with this. There would be no issue, as long as, like, things are labeled, with some sort of GUI, right? Like some sort of GUI ID that is not specific to participants. Other than that, there's no issue.
Yeah, at least for me, coming from a clinical laboratory sciences background, for us, what matters most is structure. So we label everything. We link the label to the research records. So I have to clarify that this is a research protocol. But we want to pursue or establish a clinical setting here. So we label everything. We make sure everything is at a good temperature. So validation against venous samples is the key here, and we demonstrate that we were able to do it. Really, compliance comes down to traceability, at least, and validation. So both were proved here.
Okay, wonderful. The next question that we have here, it says, in community-based studies involving pediatric or vulnerable populations, how does truCOLLECT perform in terms of DNA yield consistency and downstream compatibility with multi-omics workflows? WGS, methyl-seq, proteomics, have you validated cross-platform reproducibility compared to venous blood collection?
Who wants to take this?
Well,
I think, yeah,
You should answer.
Yeah, for me, I think, again, we proved that the biggest impact we have seen is better participation, better access. So for families, they're more willing to participate, collect at home. They prefer the finger stick versus the venous puncture. So for kids and vulnerable kids, that increase access and participation in research protocols. And we demonstrate also that for DNA-based testing, so the yield was really good. So compared to venous samples, and DNA works well. We don't have to be worried about temperature again. So this is a really good issue. We don't have to depend on frozen samples shipping, so we can take we demonstrate that we can validate that in the protocol.
Yeah, I can say that that the yields are definitely compatible with all sorts of WGS analysis, even without amplification. I mean, we used 200 nanograms for the library prep, which was average, and you can do methyl-seq, you can do all sorts of you can do a whole exome by amplification, by hybridization, and you can do the orthogonal analysis on top of that because you have enough DNA. We have done other studies with other collaborators, and they have done orthogonal analysis beyond just the WGS. But I think the WGS is the biggest barrier for a lot of other sampling methods because you don't get enough DNA out of it, or it's inconsistently in quality. And then it becomes like, you collect, you ship it, and then you stand in front of a sample that is garbage. And, all of that money is lost, and all of the use. And people you lose trust, because when your sample is lost, you participate in a genetic study for whatever reason, and your sample is lost. I mean, how likely will you participate again?
Rory, can you speak on like other applications, non-DNA applications as well?
Yeah, we have done a study actually, which was published. It's a Patrick McCarthy et al. A few years ago, where we did plasma IgG titers and compared the plasma IgG titer that we got out of the truCOLLECT. We didn't isolate plasma; we just extracted the IgGs right out of the desiccated blood, and then compared it to phlebotomy, to matching phlebotomy samples that were plasma, so where plasma was isolated. And the R squares of the SARS antibodies that we got for the N and for the S and for the other IgGs that we tested was 0.96 to 0.98 R square, and we got a higher sensitivity. We've done some other proteins, some other plasma proteins, but this is really like collaborations, because what do? We can do CRP. I mean, what people are interested in, and we can do RNA. We are beginning to do this. RNA needs a little bit more of a stabilizer in there, and of course, we cannot use anything that is toxic, because we send it directly to the patient or to the participant. So it is a little bit of a different matrix that is in there that immobilizes the blood, but we can do RNA as well, looking for collaborators.
All right, wonderful. The next one that we have here says, great presentation. What would you say are the top 3 benefits of the truCOLLECT kit?
I'm not saying anything you say or something like that. You used it.
I can say easy to manage, increased participation, and doesn't depend on changing the temperature. So it can keep in room temperature.
And I think like the key for the no biohazard, like having been in a study that shipping saliva was such a challenge and required like just so much effort on a per country basis, it's much more like this is much more feasible when dealing with multiple countries in a study.
I just want to add something really important for at least for my study. So using saliva increases DNA contamination, especially if people eat before the procedure. So that's another advantage of using truCOLLECT. So I am avoiding the contamination with other DNA, but also I am ensuring that I am using the participant's DNA and is enough to do good quality methodology.
Yeah. It's the quality and quantity that you get out of it. I think this is, for me, as the R&D guy, is the most important stuff, because I can do multiple rounds of sequencing, and good luck getting that with with the older alternatives TPS paper. I have done that, and I got frustrated because you can do qPCR, you can do a few things, but you cannot do a non-amplified NGS analysis without any barriers there.
Okay, great. The next one that we have here says, would you be using truCOLLECT for future studies?
For me, yes, definitely is a yes. I will be implementing and incorporating truCOLLECT for my studies.
Yeah, I think like we are hoping to do another 5 years of our study, and I think we're exploring to see like if this is definitely like a top tier option for us for collecting thousands of more cases across Latin America, just for ease of collection, cost benefit, and all the things that we mentioned.
Also, it is good to mention that if you are working with an adult population, you can do self-collection too. So it's as simple as, testing blood sugar in your finger stick. So it's really good. So you don't depend on going a clinical setting and use a phlebotomist. You can also do a self-collection.
Okay, great. The next question we have, it says, what is the highest, lowest temperature that the sample can withstand? And for how long is the sample stable at these temperatures?
Yeah, that's we've done quite some studies. Of course, the low temperature is usually once the sample is dry. Let's just say that once the sample is desiccated, it's very stable across a wide range of temperature. You just don't want to freeze it when it's still not dry. That's the thing, because then it may thaw during transport. So you have to think about that. But if you have it at room temperature, if you have a normal setting, you can just ship it after an hour, sitting on your table. But for Alaska, I had a question from somebody, like in an audit a while back. Can I use this in Alaska up in Point Barrow because they're up there frequently? Yes, you can, because freezing, yeah, that's fine. It's stable. We also did some tests that where we exposed the desiccated blood to 60 degrees. 50 to 60 degrees, for hours, for overnight, and it was stable. We got really good DNA out of that. So it's amazing what you can do once your blood is dry. It could sit on the tarmac. I wouldn't necessarily say yes. I would let it sit on a tarmac, on a FedEx site for like overnight or during the day. But it is stable. It's quite stable. It's amazing. We didn't anticipate that.
Okay, great. The next one that we have here says, does the cost and detail of this system allow the sampling of all newborns in a country the size of the US?
I mean, I wish we would use it for all newborns, so we can basically, we have a good product. I think yes, it can. The cost is competitive. I'm not going to do this it's definitely competitive when you compare it to other sampling methods other than TBS. We know that TBS is very cheap. It's a paper, but it also has its drawbacks. You can definitely do that. The question is, do you want to store it just for storage and for archiving and doing some qPCR later? Then I would go to TBS. If you want to do serious genetic studies, want to do WGS, WGS, yes, absolutely. Yeah, I can see it for testing all newborns. And we should do that, by the way. I'm a big believer in that. Every newborn should have their pharmacogenetic profile, because that will prevent a lot of adverse events in every setting.
Perfect. Okay, it looks like we have time for a couple more here. The next question says, how can truCOLLECT enable multiomics workflows?
I can answer that. I basically touched on it before, because yes, we can do protein, plasma protein, especially. We can do RNA, and we can do DNA. We definitely can do metabolites, small molecules. Definitely, we can extract that. We have some examples, but it will support it. The only thing what it will not support, and I will say that because very often people ask me about that, is definitely like cfDNA of very low amounts of cfDNA from cancer samples, because remember, it's 50 microliters, so you get about maximum 40 microliters of plasma out of that. But for any other studies, protein, definitely, we are very interested in finding somebody who wants to do a study with us about certain biomarkers, interested in biomarkers and doing that. RNA expression upon diet changes. What I saw recently, a paper about methylation, methyl genome or methylation changes upon diets. It's a big European study. It was published recently. So you change the diet in pediatrics, and guess what? Your cardiovascular proteins, the benefits go up. They're less methylated. The other ones get methylated and they're shut down. You get less diabetes. So these are studies that it's made for longitudinal studies, using truCOLLECT, resampling, and having people not come to the hospital for that.
Perfect. Okay, we will go ahead and close out with this final question here. So this one says, has this method been tested for protein or RNA extraction? Is it compatible with these or only DNA extraction? If I collected one sample from a participant, is there a way to rehydrate and then split the sample to extract different targets, i.e. protein and DNA, RNA?
It's a good question. I could imagine that we can split certain proteins and then DNA. RNA and DNA is difficult, and I would do a double collection with something that has a special RNA stabilizer in it. That is, that is a better idea, because don't remember, RNA you need it metered. So as long as you adulterate the sample by splitting or whatever, you don't know what you lose. I would do a RNA and DNA I would keep separate, but DNA and protein I can see something like that as something that can be done. We haven't done it though, but yes, if this is of an interest, contact us. I mean, definitely talk.
Okay, wonderful. Well, thank you all again for your time today and for your very important research. We would also like to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webinar. Now, before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions that we may not have had time for and those submitted during the on-demand period will be addressed by the speakers via the contact info you provided at the time of registration. As a reminder, this webinar can be viewed on demand. LabRoots will go ahead and alert you via email when it's available for replay. We encourage our attendees to share that email with any colleagues that may have missed today's live event. All right, thank you so much, everybody. Until next time, goodbye.
Goodbye.